In 1952, Selman Waksman was awarded the Nobel Prize in Medicine for the discovery of streptomycin, the first effective antibiotic against tuberculosis. But the award carried some controversy. One of Waksman's students, Albert Schatz, had his name on both the streptomycin patent as well as a scientific paper introducing streptomycin to the scientific community. Schatz had a legitimate claim as the co-discoverer of this antibiotic that treated the deadliest disease in human history. It would have been nice to be acknowledged. So I wanted to know, why was streptomycin's discovery such an important moment? How did these scientists actually find it? And finally, what happened between Waksman and Schatz? In the 1880s, Robert Koch identified a bacteria called Mycobacterium tuberculosis as the cause of the disease tuberculosis. Afterwards, improved hygiene and a better understanding of the disease's transmission helped reduce the incidence of TB, at least in the United States. But even into the 20th century, there weren't any great treatments. There was a vaccine for tuberculosis, but it wasn't very effective. By the 1940s, we had a few antibacterials for other infections. There were sulfonamides, which hit the scene in the 1930s, and beta-lactams like penicillin in the early 1940s. Unfortunately, neither of them worked well against Mycobacterium tuberculosis. They also didn't work against the germs that cause cholera or the plague, but tuberculosis was the bigger concern. So why didn't they work? Why was TB so special? Well, despite a prefix that might make you think it's a fungus, the myco in Mycobacterium tuberculosis refers to the mycolic acid in its cell wall, and that mycolic acid makes this thing really tricky to treat. Up until this point in the series, I've mostly talked about two big camps of bacteria. There are gram-positive bacteria, which have thick cell walls made up of a substance called peptidoglycan, and there are gram-negative bacteria. These have a much thinner layer of peptidoglycan that's surrounded by cell membranes. Beta-lactam drugs like penicillin work by disrupting the formation of peptidoglycan, but they can't easily cross the cell membrane, so they're much more effective against gram-positive bacteria than gram-negative bacteria, like the ones that cause plague and cholera. That's why penicillin wasn't useful against them. Now, Mycobacterium tuberculosis does have peptidoglycan in its cell wall, but that layer of waxy mycolic acid prevents penicillin from reaching it. The other major class of antibacterials at the time was sulfonamide drugs, which keep the bacteria from making folate, a chemical it needs to replicate. And doctors in the 1930s had some success using sulfa drugs against TB, but they weren't very effective. And again, all of this is set against the backdrop of the fact that tuberculosis is the deadliest disease in human history. This is where Selman Waksman comes in. Waksman was a soil microbiologist at Rutgers University, and he was especially interested in a type of stringy microbe called actinomycetes. These little guys are weird. Scientists used to think they were fungi because their anatomy is really similar to fungi, but they're actually bacteria, and Waksman made them his life's work. In 1923, Waksman and his student, Robert Starkey, noticed something. Certain types of actinomycetes could kill other bacteria. Kinda like how penicillin left a ring of dead bacteria around it, actinomycetes was often the bullseye on a target of dead microbes. So, Waksman's lab spent the 1920s and 30s collecting soil samples around the eastern United States, picking out the actinomycetes, and learning everything they could about them. They were convinced that there was something special about these microbes that could keep other microbes from growing. And in the process, Waksman churned out some interesting students, one of which was a French scientist named René Dubos. And in 1927, he became a researcher at the Rockefeller Institute. He actually ended up working under Oswald Avery, who I talked about during the Spanish flu vaccine video. Avery got Dubos to study how soil bacteria might break down the protective capsule around a specific type of pneumococcal bacteria. After a while, he isolated an enzyme that he called S3, and by 1930, he'd used it to successfully cure mice with pneumonia, and it looked like it might be a useful antibiotic, but before he could make a version of the medicine for humans, the Germans came out with the first sulfa drugs in 1935, and those got way more popular. But it was an encouraging start, because it showed that soil microbes could be a potential source of antibiotics. And in 1939, Dubos published a series of papers describing another potential antibiotic he found. The first paper in the trilogy showed that the substance destroyed cells in vitro, meaning in the petri dish. Paper number 2 showed that it protected against pneumococcal infections in vivo, meaning in live mice, and paper 3 described what the antibiotic actually was, and how to extract it from the bacteria. No human tests yet, but it looked promising in mice. This substance, tyrothricin, was made by a bacteria called Bacillus brevis, And in 1941, Dubos released a paper saying that tyrothricin was actually made of two different antibiotic chemicals, tyrosidine and gramicidin. Now, much like penicillin, these antibiotics were much more effective against gram-positive bacteria than gram-negative bacteria, although with different mechanisms than penicillin. Tyrosidine keeps a bunch of important proteins from forming inside the cell, and gramicidin creates little pores on the surface of the bacteria. This makes the cell leakier, which throws off the fluid balance and breaks open the cell. Unfortunately, both of those antibiotics were just too toxic to be useful for systemic human infections. Gramicidin actually is still sometimes used as a topical treatment, but we wouldn't be able to safely inject it into someone's veins to cure sepsis or something. Either way, Dubos had properly shown that soil microbes could produce medicine, and this encouraged other researchers to keep looking for antibiotics, especially Waksman, but also the Oxford team who produced penicillin. Plus, at this point, it's World War II, so if you're going to find some antibiotics, now's a good time to do so. In 1939, the same year that Dubos came out with his research, Waksman started a business relationship with the pharmaceutical company Merck. See, Waksman's lab was made of microbiologists, and they didn't have the licensing or resources to conduct animal testing in-house. So, as part of their partnership, Merck would supply those resources plus chemicals and equipment for large-scale production if any of their antibiotics looked promising. In return for those resources, Merck would get the patent on any antibacterial products that came from the research. Obviously, this was a great deal for them, but they also agreed that if any of those antibiotics were commercially successful, they would pay a royalty to the Rutgers Foundation, a nonprofit entity. The relationship started in 1939, and it didn't really return anything to Merck for the first year. But in 1940, a graduate student named Harold Boyd Woodruff published his findings on a potential winner. He and Waksman named this compound actinomycin, and its bacteria Actinomyces antibioticus. Later on, Waksman worked with a chemist at Merck named Max Tischler to study its chemical structure, and this is the resulting patent. You're starting to see how the relationship is going to work out. Actinomycin worked against a couple pathogens in vitro, mostly gram-positive ones, but it was just too toxic to be useful in humans. And that was a bummer, but it also lit a fire under the Waksman team to start a whole new process of microbe hunting. They'd perform a meticulous screening process for soil microbes so that they could find good antibiotics. At first, they'd put the Actinomycetes on a plate of agar alongside different pathogens, bacteria that cause disease. Then they'd measure the size of the ring around the microbe. Bigger ring equaled a stronger antibacterial effect. In the next step, the microbe needed to produce the antibiotic compound in big enough quantities that it would be practical to produce them. And finally, it had to be safety tested. Of the thousands of microbes screened, this process yielded about 20 new antibacterials, most of which came from Actinomycetes. It's around this time when Waksman coins the term antibiotic. It was a substance that killed bacteria secreted by another microorganism. I'll give you a quick example. The syphilis drug Salvarsan doesn't count as a true antibiotic, since it was synthesized in a lab, but penicillin does count since it was made by Penicillium, which is a mold. Speaking of which, penicillin started to roll out to its first few dozen patients around mid-1942, and it was becoming clear that it was better at treating gram-positive bacteria than gram-negative bacteria. That's why the Rutgers team was so excited to isolate streptothricin next, which seemed promising against gram-negative bacteria in particular. Unfortunately, this one ended up being toxic in humans too, so no dice. They had two more maybes in 1942, clavicin and fumigacin, but they had the same problems, toxic in vivo. But 4 near hits looked pretty good, so they kept going. That year, Waksman's lab hired a fresh grad student named Albert Schatz. He actually got drafted to the army for a couple of months, but was honorably discharged in June of 1943. He would test antibiotics on a few gram-negative bacteria, including E. coli and a type of mycobacterium that doesn't cause disease. Later on, he started experimenting on the real deal, Mycobacterium tuberculosis, at which point Waksman moved him into the basement for obvious safety reasons. And pretty soon into his research, Schatz came across a microbe called Actinomyces griseus, a microbe that Waksman had previously studied. Between September and October, he actually got 2 strains of it from 2 different sources. One of the strains came from a sick chicken isolated by poultry virologists at Rutgers, and the other came from a regular soil screen. Although Waksman wondered if this second strain was really just a soil sample that had been contaminated by the first strain. The antibiotic substance turned out to be effective against both gram-negative bacteria and gram-positive bacteria, including, importantly, the germ that causes tuberculosis. They named this substance Streptomycin. Along the way, they renamed the microbe Streptomyces griseus. Waksman actually wrote a whole paper about this renaming. It's a little too nerdy for this video, so I'll link it down in the description with all my other sources. Streptomycin was the first in a class of antibiotics called aminoglycosides. Usually you'll know these drugs because they end in mycin, or just sin, like the neomycin that you might know from Neosporin is an aminoglycoside. These kinds of antibiotics ultimately kill bacteria by keeping the cell from making proteins. But to understand how it works, we need a quick biology lesson. So big picture, the DNA in our cells is a set of instructions for making stuff, mostly proteins. But the cellular machinery that makes proteins, the ribosome, can't read DNA directly. So we need to turn it into a form that it can read, what's called messenger RNA, or mRNA. And the first step in this process is called transcription. In this step, enzymes use DNA like a template to create matching mRNA, and that mRNA can be read by the ribosome in a process called translation. Now, all around the cell are little molecules called tRNA, or transfer RNA. These guys are kind of like dongles for your computer. On one end is a slot for a specific sequence of mRNA, and on the other end is a slot for a specific amino acid. For example, if this sequence of mRNA says AAG, a tRNA will bring the amino acid lysine to the ribosome since that's what AAG codes for. Ribosomes themselves have two parts, a small subunit and a large subunit. The small subunit is where the ribosome reads the tRNA, where the right tRNA brings the right amino acid. Then the large subunit links the amino acids together into a polypeptide chain. This is where the antibiotic comes in. Aminoglycosides like streptomycin bind to the small subunit, which keeps it from putting the right amino acid in order when a tRNA brings it one. So instead of reading the genetic instructions correctly, the ribosome misreads the instructions and puts together a nonsense protein that harms the bacteria. It's also been shown to block the initiation of the protein building process, but either way, the result is messed up protein synthesis and death of the bacteria. Okay, so back to Rutgers in the fall of 1943. This was all really exciting. The drug destroyed Mycobacterium tuberculosis in a petri dish, but they'd eventually need to test it on animals. Coincidentally, in November of 1943, two infectious disease researchers from the Mayo Clinic got in touch with Waksman for a collaboration. They were William Feldman and his intern at the time, Corwin Hinshaw. These two were especially interested in the anti-tuberculosis effects of clavicin, but they knew it was too toxic to use. So the Mayo guys are like, hey, give us a ring if you ever find something interesting. We might be able to work together. Now, like I mentioned, Waksman needed to find someone to do animal experiments anyway, so he kept them in mind. Fast forward to January of 1944. Schatz, Waksman, and another researcher named Elizabeth Bougie published their first streptomycin paper in the Journal of Experimental Biology and Medicine. Then in March, Waksman asked the Mayo boys if they'd be willing to do clinical testing in living subjects. Next, he had Schatz pump out the antibiotic in his basement lab, and streptomycin testing at the Mayo Clinic could officially start in April. By May, they had good evidence showing that streptomycin wasn't just safe, but could cure tuberculosis in guinea pigs. And by June, it was clear that streptomycin cured different gram-negative bacteria too, like the ones that cause plague and tularemia in test animals. It was looking good. These were exciting initial results, but they would need a large sample size and proper controls before they moved on to human trials, and that meant a lot more streptomycin production. Luckily, Waksman had that consultant relationship with Merck, so they would have been the natural partner for streptomycin production. But we need to remember the timeline. This was 1944, when Merck, as well as most major American pharmaceutical companies, were all in on penicillin production for the war effort. They couldn't risk their resources on an unproven drug when penicillin was so valuable. And even if they had the capacity, gram-positive infections like staph, strep, and tetanus were some of the most common infections in soldiers, and streptomycin didn't help against those infections. But despite the circumstances, the Mayo boys asked Waksman to introduce them to Merck. At the very least, maybe they could convince them to get them enough resources so they could continue testing. Now, in this meeting is Merck's president, George Merck. He saw the future potential of the drug and decided to invest resources into streptomycin development after all. A month later, Merck started construction on a new facility in Virginia that would be dedicated to streptomycin production, and he assigned a team of researchers to the project. And in the meantime, the Mayo Clinic finally got enough streptomycin for in vivo experiments. They infected 49 guinea pigs with Mycobacterium tuberculosis, then gave half of them a series of streptomycin while the others acted as controls. 18 of the 24 control animals developed tubercles and 17 died. Meanwhile, only one animal in the drug group developed tubercles and only 2 died. But over the summer, there were some issues with toxicity, which might have been due to contamination in the antibiotic. But by August, they were starting to get some of the first Merck versions of streptomycin, which were more pure and less toxic. Now they were ready for human trials. The first case was actually a 2-week-old infant with meningitis, septicemia, a blood infection, and a urinary tract infection. Sulfa drugs and penicillin got rid of the blood infection, but the UTI wasn't responding, so they gave the baby streptomycin over the course of 6 days and it cleared the infection. This was great. Streptomycin showed that it was safe and effective in a human, but everyone was looking forward to seeing how it would do against the big one, tuberculosis. That patient would be a 20-year-old woman named Patricia Patsy Thomas. She'd spent over a year in a tuberculosis sanatorium and was getting worse. So in October of 1944, she got sent to the Mayo Clinic for a consult with Hyeon-Seo, who started her on a regimen of streptomycin over the course of 5 months. The tricky part was figuring out dosage. They had a good idea on best dosage based on guinea pigs, so they scaled up the dose based on their patient's weight, and by April of 1945, her TB had been cured, and she lived for another 21 years. By September of 1945, the Mayo Clinic doctors had treated over 30 patients with TB and published their results in the Proceedings of the Mayo Clinic. Streptomycin was granted FDA approval for the treatment of tuberculosis in 1947, and we'll get into the impact of this drug, but now comes the messy part: money and fame. In the United States, you can't patent a, quote, natural product. By definition, antibiotics are made by bacteria, so you'd think they're a natural product and therefore unpatentable, right? Merck got around this for actinomycin's patent with some creativity. Waksman was able to convince patent officers that their lab had purified, transformed, and prepared the chemical into a form that doesn't exist in nature and should be patentable. They used the same strategy for streptomycin. On February 9th, 1945, Waksman and Schatz file a patent as equal, joint, co-discoverers of streptomycin. While Elizabeth Bougie's name is on the original streptomycin paper, she claimed that she didn't really develop anything novel and she shouldn't be on the patent. So they filed the patent, but for some reason, Waksman wasn't comfortable with the idea that the rights to streptomycin would go exclusively to Merck, as per their arrangement. And you can see why. This drug had the potential to cure tuberculosis and plague. It should be mass-produced and cheaply available. So Waksman approaches Merck, and they agreed that streptomycin would do more good with a non-exclusive patent. Merck agreed to sell the patent back to Rutgers. Not for free, but they did let go of some serious potential profit, something they like to highlight on their website. Now, nobody knows exactly why Merck agreed to the deal, but it probably had something to do with World War II. The president of Merck, George Merck, was the head of the War Research Service and was worried that the Axis powers might use biological weapons, so he decided to forego profits on streptomycin in order to get more companies to produce it, giving the Allies a better tool against their enemies. This is the guy who got the cover of Time magazine in 1952 with the quote, "Medicine is for the people, not for profits," which is a quote that would sound bizarre from pharma company leadership in modern day. But before they could transfer the patent, Waksman asked Schatz, "Hey, are you cool if we don't make any money off this?" And Schatz agreed. He had every reason to trust his mentor. They sold their patent to the Rutgers Research and Endowment Foundation for $1 apiece. Now, from a public health perspective, this was awesome. Rutgers licensed out the patent to multiple drug companies, which meant more streptomycin got out there and doctors finally had an antibiotic that could treat tuberculosis. But from a business perspective, the patent transfer is just the beginning. After filing the patent in 1945, Schatz earned his PhD and continued working on streptomycin production at Rutgers for another year. And at this point, things seemed totally cordial between him and Waksman. The supervisor wrote his student a letter of recommendation for a job, which he got, and Schatz wrote him a nice thank you letter. Totally fine. Then for most of 1946, Waksman had the best professional year of his life, winning all kinds of prizes and awards. But over the course of 1948, newspapers were making it seem like Waksman was the sole discoverer of streptomycin, and the next year, he got a profile in Time magazine with no mention of his research assistants. The patent was finally granted in 1948, at which point, the drama really gets started. Schatz started to hear rumors that Waksman was collecting some royalty checks from the streptomycin patent, which struck him as weird considering they sold it to the Rutgers Foundation. So in January 1949, Schatz wrote a letter to Waksman asking him what's going on. It turns out that while they did sign their patent over to the Rutgers nonprofit, Waksman was getting paid from the nonprofit. For every dollar that the Rutgers Foundation made from licensing out streptomycin, Waksman got 20 cents. By 1950, he'd collected about $350,000 in royalties. Now, a lot of that royalty money went towards funding streptomycin research, but yes, some of it did go towards his salary. It's not clear how Schatz found out about this, but needless to say, he was heartbroken. He trusted Waksman and signed away his patent with the understanding that neither of them were going to profit. For the next couple months, they wrote letters back and forth to each other, with Schatz saying that he deserved payment as a joint discoverer of streptomycin, and Waksman saying, "Yeah, but it was my lab." The most gut-wrenching line from their exchanges has got to be from Schatz to Waksman. You once told me that in certain respects you have been as a father to me. Since this is true, it is in a sense, therefore, that I am writing to you as a son. I have every confidence that you will advise me as a father to his son. Schatz sued Waksman in March of 1950, and the case was settled by December of that year. The result was that Waksman would go on to keep 10% of the royalties, with 3% going to Schatz and 7% to the remaining people who worked in the lab during the research's high time. Schatz also sold any future foreign licensing rights for a total of $125,000. 2 years later, Waksman alone was awarded the Nobel Prize in Medicine for the discovery of streptomycin. A representative from Schatz's workplace reached out to the Nobel Committee on his behalf, but nothing ever came from it. And this brings us back to the opening controversy. The idea of scientific discovery is messy. It's so rarely just one scientist. It's always more likely a team, especially in modern day. And that ignores the historical basis. You've all heard of the phrase, "I stand on the shoulders of giants." These days, people still have hot takes about Waksman versus Schatz, and there are basically two takes. The pro-Schatz fans say that he did something scientifically novel that deserves credit. The pro-Waksman camp says that he did the decades of institution building and screening and hard work and basically set up his student to get lucky. Let me know your takes in the comments. I'm ready to move on and talk about the impact of the drug. During reading, the phrase I saw the most was that streptomycin, quote, filled in the gaps created by penicillin. According to Selman Waksman's obituary in the British Medical Journal, it is almost impossible to exaggerate the importance of this discovery. Streptomycin not only filled a large number of gram-negative gaps in the spectrum of penicillin, including efficacy in plague, but proved effective in the treatment of tuberculosis. Patients even began to recover from tubercular meningitis, until then the most inevitably and regularly fatal of all the bacterial infections. But even with this awesome new antibiotic, there were still some big questions. Like, beta-lactam drugs had really dramatic effects. You could clear a staph infection in a few days with penicillin, but streptomycin took months to clear TB. So how are they sure it even did anything? Well, starting in 1947, the UK's Medical Research Council, or MRC, recruited over 100 young patients with tuberculosis to be part of a study. Half of the participants were randomly assigned to bed rest and streptomycin, and half of them were assigned to bed rest and a placebo. Each participant got a chest x-ray at the beginning and end of the trial, and a radiologist evaluated it. What was cool and kind of novel for the time was that the radiologist didn't know what kind of treatment the participant got, so they could make their assessment without bias towards or against the medicine. This was actually one of the very first double-blind studies on record. Neither the participants nor the assessors knew what treatment each participant received. And on first glance, the results seem pretty clear. 14 of the original 52 controls had died after 6 months, compared to 4 of the 47 who got streptomycin. Unfortunately, it took a long time before you saw any meaningful difference between the two trial groups, and that was enough time for TB to build resistance to streptomycin. Case in point, when they followed up 3 years later, the death rate between both groups was almost equal. Luckily, scientists came up with a clever workaround. A Danish doctor named Jørgen Lehmann, I'm almost certainly butchering that, knew that you could increase Mycobacterium tuberculosis' oxygen consumption with a chemical called acetylsalicylic acid, the active ingredient in aspirin. So he tested a bunch of similar compounds, and in 1946 found that para-aminosalicylic acid, or PAS, was the most effective of them. And PAS was fine. It wasn't a miracle cure, but it had some effect against TB. But shortly afterward, the first group of researchers in the UK wondered what would happen if you combined PAS with streptomycin, so they ended up conducting a similar trial to their original one. And this trial showed a much stronger effect size when the two drugs were combined, 80% survival rate after 3 years. Longer term, the addition of PAS led to less toxicity too. Got it. Streptomycin plus PAS was the way to go. Of course, scientists have developed more aminoglycoside antibiotics since streptomycin, with probably the most popular one, gentamicin, getting developed in 1963. Although these days, the current standard of care is the RIPE combination therapy, which stands for these drugs. None of them are aminoglycosides. There are other treatment regimens out there for TB too, like rifapentine, rifabutin, linezolid, and some fluoroquinolones. Streptomycin might be useful in cases where RIPE therapy isn't available, or it might be used in combination therapies when the bacteria is resistant to RIPE. But of course, in the 40s and 50s, this was all great news. Not only did we have penicillin for gram-positive bacteria, but now we had PAS plus streptomycin for TB and gram-negative bacteria. But what if we could find an antibiotic that fought both kinds of bacteria? Whoever created that antibiotic would make a lot of money indeed. Next up, the story of the first tetracyclines.